Hi students, good morning and welcome again to this class. Alright, we are now rejuvenated because of the festival break. We enjoyed a lot. Let us now concentrate in our studies again. So about the sensory organs in human, eye and ear. I had started in my last video. So today what I am going to do, I am going to elaborate a little more, which are important for you students in class 11. So when you talk about eye, the eyeball comes in mind, which I have discussed already, started discussing in my last video, and the other parts associated with eye you are already aware of, like the eyebrows, the eyelashes, the eyelids, and the lacrimal glands. Where are these lacrimal glands located? They are at the outer upper end of the eyeballs. What are they doing? They are secreting tears. Fine, tears are there for communicating emotions, okay, alright. But they are doing some other important functions as they are destroying the germs which can enter into the eyes. How they are doing this? Because they are having lysosome, an enzyme, and bactericidal enzyme which is directly killing the germs. Bactericidal means directly killing. Bacteriostatic means, as you are all aware of, bacteriostatic means inhibiting the growth and then killing the germs. But lysosome is bactericidal, so it is directly destroying the germs, right? And what else the tears are doing? They are lubricating, cleaning the eyeball, and when we blink, it is spread around the eyeball. So these are the functions of the tears that is secreted by the tears secreted by the lacrimal glands. Okay, the location and function of the tears you have to remember. Location of the lacrimal glands and function of the tears. Now about the eyeball as such, what is the size? It is about 2.5 centimeters. But what part we are seeing is only 1/6 of the frontal part of the eyeball. Then the rest of the eyeball, what happens? The rest of the eyeball is embedded in the bony cage. Now how this eyeball is there at its position? How it is being kept in its position? It is kept in its position by 6 muscles which are attaching eyeball to the bones, right? So these are the important features that you have to remember, students. Now when we talk about the other eye parts, you are already aware, you are already studied about the, I have already discussed in my last video, the sclera, steroid or the choroid and the ciliary muscles and the retina. Okay, so retina, when you talk about, you must remember the sensitive cells present in retina, which are enabling us to see actually the in dim light and also in bright light. In dim light we are able to see because of the rods, which are the sensitive cells, which are having rhodopsin, right, as an enzyme. And in bright light we are able to see because of the cones. Where they are situated or located, they are found in the center of the retina. So maximum concentration of cones are there in the center of the retina, and we are able to see in the brightness because of these cones. We are bright light we are able to see, which is called the phototropic vision. Phototropic vision is seeing in color. The color vision we are able to to have a color vision. Cones are specialized for color vision and sharpness of vision in bright light, which is called phototropic vision. So these small but important words you have to remember, students. And what are these dark spots and the blind spot and the yellow spot? Yellow spot and blind spot also you have to remember. Yellow spot, rods are concentrated towards the periphery, while the cones are concentrated, as I said, at the center on the horizontal axis of the eyeball. At the center, it is they are located. This spot, which has the greatest concentration of cones, is called the yellow spot, because the cones are in maximum number at the center, and these highly concentrated concentration of cones makes it the region of brightest and sharpest vision. Region of brightest and sharpest vision is where? At the yellow spot, where the cones are at the greatest concentration. So naturally, the cones are enabling us to see in bright light. So maximum vision will be here only. So that's it. That is why it is called the area of maximum vision. This yellow spot, region of best vision or maximum vision. It is called region of the best vision. And blind spot, region of no vision. There is an also just below the yellow spot. There is an area where there is no rods, no cones. Only openings are there through which the axons of the optic nerve is coming out. So there is no enzyme, there is no sensitive cells. That is why there is no vision, because these sensitive cells are, enable us to see. So if they are absent, there will be no vision. So that is why this spot is called the blind spot, and this you have to remember, both the yellow spot and blind spot, right? And now other things which you have to just revise and remember is the mechanism of vision and what is happening. The light rays in visible wavelength focused on the retina through the cornea and lens that you all are aware of generate potentials. What is the potentials? These are the impulses. You have already heard in the last chapters the resting potential, the action potential, and here which take place, which take part in the conduction of the impulses. So here also these potentials are generated in rods and cones. These are the sensitive cells. As mentioned earlier, the photosensitive compounds in the human eyes is composed of, what is the photosynthetic compounds, photosensitive compounds which are sensitive to light? They are composed of proteins only. So all these enzymes, these rods are containing rhodopsin, the cones are containing rhodopsin. Enzymes are what? They are proteins only. You just remember. So these the proteins. In a human eye is composed of opsin, which is also a protein, and retinal and aldehyde of vitamin A. These opsin and retinal, these are the proteins in the sensitive cells, rods and cones, right? In the enzymes, rhodopsin and rhodopsin. So these retinal and aldehyde of vitamin A, light induces dissociation of retinal from opsin, resulting in changes in the structure of the opsin. This causes membrane permeability changes. As a result, potential differences are generated in the photoreceptor cells. This produces a signal that generates action potentials in the ganglion cells through the bipolar cells. These action potentials are transmitted by the optic nerves to the visual cortex area of the brain, where the neural impulses are analyzed and the image formed on the retina is recognized based on earlier memory and experience. So what is happening actually, students, you have to understand very clearly in the mechanism of vision is, see, these are the proteins which are there in the sensitive cells, rods and cones. Which are the proteins? Because all the enzymes are made of proteins. the proteins are opsin and retinal, right? And retinal is what? An aldehyde of vitamin A, right? So protein is there with a aldehyde of vitamin A. So these two are separated. Light, how? By the light. Light induces dissociation of the retinal from opsin. So this dissociation takes place, separation takes place by the light of this vitamin A aldehyde, that is the retinal, from the protein opsin. So this separation takes place because of the light, or dissociation takes place, which results in what? In changes in the structure of opsin. So there will be a change in the structure of the protein, that is opsin. So these changes will cause what? These causes membrane permeability changes. So membrane in the neurons, in the neuron, the permeability will change. So potential differences will be generated whenever there is a change in the permeability. The potential difference takes place. The difference in concentration of the ions takes place, sodium and potassium ions. So this gives rise to potential differences are generated where? In the photoreceptor cells, because in the photoreceptor cells all these things are happening. Neurons are there. So in the ganglion, neurons are there. So these changes are taking place there. This produces a signal. This produces what? A signal that generates action potential. This potential difference will generate a signal. That is what? That is the action potential. So because of the change in the permeability of the membrane, the transport of the ions will differ. There will be again a change in the concentration of the ions. So this is called the difference in concentration, and this causes membrane permeability changes. As a result, there will be a difference in concentration, which is called the potential difference. I had talked earlier also, and these potential differences are generated in the photoreceptor cells, right? This produces a signal. This potential difference will produce a signal that generates action potential. That will generate another impulse, that is called the action potential. So this concentration difference will generate a potential which will potential differences which will be there in the photoreceptor cells. This potential difference will result into another action potential, which is what? Which is the impulse actually, and this produces a signal that generates action potential in the ganglion cells through the bipolar cells. These action potentials or impulses are transmitted by the optic nerves to the visual area. There is a visual area in the brain, and then the correct interpretation takes place, and we are seeing the objects as per our earlier memory and experience. So this is what is happening in the mechanism of vision. This you have to remember in class 11. Students, I am ending this video, and again I will, before it becomes too long, I will start the year chapter in my next video. So I think this will help you. These tips or these pieces will help you in your preparations. See you again. Till then, goodbye, and God bless you all. If you like my videos, please do subscribe to my channel. Okay, thank you very much. God bless you all.